That's not the first line. Okay, so I was kind of writing on this before. And so this is sort of the standard Mendelian um, crosses that we talked about when we were looking at dihybrid crosses. And so you remember we could have the seeds that were green or yellow, and yellow was dominant. Um, this is using G, which is a weird nomenclature, but it doesn't really matter regardless. And then remember they were round or wrinkled, and so in this case, round was dominant to wrinkled, so we use R. They're using W, which is completely backwards of what we had discussed before, but again, it's fine. So, so we know that there are two uh, different phenotypic traits that come out of this, and it's either P shape or P color. And this gene has no effect on the P shape, right? Because it's the color gene, and in this case, it's green or yellow. And this particular gene, um, it has no effect on P color, only shape. So these genes are independent of one another. They don't interact in an epistatic way, which we had already shown before in the, I think in the first lecture. Here's this menu again. Move it. I don't know if you can see this or not, but I can. Okay, so in reality, almost every phenotype is affected by many genes in a pathway. Um, when Mendel did his experiments, they were really weird. A lot of scientists questioned that whether they were actually um, real, and that if you put did he put all the data in there or not, or was he just really crazy lucky that all of the genes that he had studied were independent on separate chromosomes and followed the standard ratios? Because it's quite likely that he did some experiments that did not follow his data, and he just basically tossed that. So generally, and I was just talking about this in my 181 class, when we're looking at pathways like uh, glycolysis, for example, and hopefully this is a review for 181. So you have glucose, and glucose enters the cell. So you eat your food, and your saliva, the amylase in your saliva, digest amylose, which you would see on your nutritional label as carbohydrates, and that breaks down the starch, right? The amylose, same thing, into maltose. So amylase will bind to and cause a dehydration reaction for amylose, and we end up with two hexose sugars linked together, and this is called maltose. And this is a really important molecule. That molecule is used uh, quite frequently in the production of beer, so it's a multi-billion-dollar molecule here. And so when you digest that, your stomach breaks this glycosidic linkage further, and you end up with glucose. And that glucose goes through your small intestines and enters your blood, where it causes insulin to be secreted by the pancreas, and then that insulin binds to your cells. So here's your cell, here's insulin, binds to that cell, sends a signal, opens the channel, and then glucose enters. All right, so now we have glucose in the cell, and then the glycolysis occurs. So you should have learned this in introductory biology for majors, and if you didn't, then you probably should talk to your teacher about that. So glycolysis is a pathway where we have glucose. We start with glucose, and then glucose gets phosphorylated on the number 6 carbon by kinase, and so it becomes glucose 6 phosphate. Your teacher may have not gone in this depth with you. But anyway, and so that gets rearranged by an isomerase. Hopefully, you know what an isomer is, and hopefully, you learn that in intro biology. And then that isomerase converts glucose into fructose, 6 phosphate, and then a fructokinase adds another phosphate in the 1, so you get a 1,6 fructose. And then in that pathway after that, it gets split into 2 by another enzyme. So every one of these enzymes that I'm talking about, like this kinase, this isomerase, here's the kinase, right? That adds a phosphate. Isomerase is rearranging it. We have another kinase, so it's called a fructokinase, and that kinase is going to add another phosphate to the number 1 carbon. So now we get a 1,6 fructose. So every one of these enzymes are in this pathway, right? And so this one, when we're talking about the pathway, this is upstream, right? So the one that happened first, and in this direction it would be downstream. So if I were to continue this, there's a bunch of other enzymes, and then it ends up being glyceraldehyde 3-phosphate, and that gets converted through a series of enzymes. I think there's about 6 enzymes into pyruvate, and then pyruvate gets put into the Krebs after it's converted to acetyl-CoA. So anyway, if we're talking about the enzymes at the beginning of glycolysis, that's upstream. If we're talking about the enzymes at the end of glycolysis, that's downstream. Okay, so let's talk about these plants. They're called harebells. Sorry, harebells. And this pathway creates a purple pigment, which makes these flowers purple. So we have a enzyme A, right, that takes the precursor protein. It's white before this enzyme gets a hold of it, and then this enzyme causes a reaction. I don't care that you know the reaction, right? So maybe it adds a phosphate. Who cares? And so, I mean, if you were interested in this as a geneticist, you might care, but for illustration purposes, it doesn't matter. So this enzyme is going to do something. It makes another precursor, right? So this is a different protein that has a different shape because that enzyme is going to do something to it, right? Like if we add a phosphate, if I had a Lego car and I added a Lego brick to the top of it, it would have a different shape. If I took a Lego brick away from it, it would have a different shape. So a kinase adding a phosphate, so that, where's my cursor? Oh, my pen's on there. It's not going to do that. So if we have a kinase, right, it's going to add a phosphate. This glucose 6-phosphate has a different shape than glucose. And so that the example here that we're talking about is that this enzyme has done something to this precursor protein to make it a new protein with a different shape. So we're going to just call that precursor 2. And then there's another enzyme in this pathway, like isomerase, is the example that we used in the real world. And that is going to take this precursor and do something to it. So it could isomerase it or add or take away from it or, whatever. And that's going to change, it's going to change the shape of the protein. And that protein is going to fold into a specific shape. And we talked about how proteins fold. And when it does that, it's going to transmit to your eye the frequency of light, the wavelength of light that's in the purple spectrum. So that's probably 450 to 400 nanometers, somewhere in that range. And you're going to see that flower is purple. And if this enzyme didn't work, then this wouldn't get made, right? And obviously, enzymes are very specific for what they react with. So this enzyme is only going to fit on precursor 1. This enzyme is only going to fit on precursor 2. So this enzyme cannot affect precursor 1. This enzyme cannot affect precursor 2. Because like if we look at enzymes, if we look at glucose, well, that's a bad example. Let's just do a standard disaccharide. So let's say we have sucrose. So the enzyme that works on sucrose is called sucrase. And sucrase is not going to work on fructose or lactose, right? So lactose is broken. That sugar is broken down by lactase, the enzyme in milk, and so actually, if you look at lactose-free milk, it's literally had this enzyme added to it to pre-digest it, the lactose. So anyway, the point I'm trying to make is that lactase is only going to break down lactose, and sucrase is only going to break down sucrose, and fructase would only break down fructose. And these sugars are very, very similar in shape, but they're not identical. And so enzymes are very specific for what they react on. Um, they they're not gonna if they don't fit on that substrate, they're not gonna react with it. Okay, hopefully that's just review. So let's say we have a loss of function mutation, and let's say that it happens in the enzyme A, right? So we're just going to call this little a little a. That means that it's not going to make half the amount of this enzyme. It's going to make zero of enzyme A. And because there's no enzyme A, well, we're never going to get the precursor 1 to be precursor 2. It's always going to be 1. And enzyme B cannot work on 1. So because of that, we're never going to get the purple color. So in the end, if enzyme A is broken, we get white flowers. Okay, now we could have a mutation in B, and we'll call this one B, enzyme B. So here's our mutation in B. A is fine. It could be either homozygous dominant or heterozygous. As long as we have one copy, we're going to make half the amount of enzyme A, and we get half the workers. They're still going to do this job, although it might be slower. And we go from here, so we're going to get precursor 2 made, but precursor 2 can't be altered by enzyme B because there's no enzyme B, right? It's homozygous recessive, and that means no enzyme B is made, and so therefore we never get the purple color, and then in the end we get white as well. So if we're looking at these flowers, can we tell if the mutation is in A or in B from the phenotype? And the answer is no, because they're both white. It wouldn't matter. And sometimes you have pathways that are 16 long. One of those pathways has to do with Roundup. So I know this is kind of a tangent, but plants can only synthesize glucose. That's all they can make. They're not getting any proteins or anything out of the soil. If they did, then Bear Grylls would just eat dirt. There's like 5 enzymes in this pathway to convert glucose into an amino acid, right? So we have amino and carboxylic. Sorry, this is up there. Another carbon. That's the central carbon. We have another carbon that's double bonded to oxygen. This one's a single bonded to oxygen. That's hydrogen. So this is carboxyl amino. We have the hydrogen, and then we have the R groups. So at some point we have to add nitrogen, and there's going to be isomerases that rearrange that. And so we're just going to say that enzyme C, this enzyme is part of this pathway. And so if C broke, think of it as an assembly line. Like, let's say there's a bunch of workers making cars or making phones, and the workers, all the workers that knew how to put on the screens on an iPhone, walked out or walked off the job. So they weren't there. You wouldn't get any phones made because that would stop your assembly line right here. So if this enzyme C is messed up or affected in some way, you're not going to get amino acids, and so the plant can't make proteins that it needs to live, right? Because it only has glucose. It has no way to get proteins unless it's like a Venus flytrap. And in fact, they're just really trying to get nitrogen. So this pathway is affected by an enzyme, not an enzyme, a chemical called glyphosate that blocks this enzyme in this pathway. And so when you spray Roundup, which is the weed killer, on a plant, what you're effectively doing is removing one of these enzymes from its pathway, and so that shuts it down, and it can no longer make proteins, and if it can't do that, it dies. So that's how Roundup works, is it's blocking one of the enzymes in this pathway. And if you want to overcome that, then you just, like, if all your workers left and you had no one to put screens on your phone, how would you make sure your factory kept running? Well, you just hire more workers. So Monsanto inserted genes for C, more genes for C, into their Roundup resistant plants. And so we have more workers, so they just bypass this, right? They don't need, if they're, you spray, like, let's say you spray 10 units of glyphosate on there, well, you're going to get rid of 10 workers. So how do you overcome that? Well, you put another 10 workers somewhere else in the genome, or maybe 20, or maybe 30. And the more you add, the more resistant. It's not proof, because if you dunk the whole plant into glyphosate, it would eventually die. There wouldn't be enough workers to overcome the amount of glyphosate it was being exposed to. But anyway, that's basically how it worked. So that's, we're talking about the same thing. By the way, this made Monsanto like billions and billions of dollars, just knowing these pathways and knowing how to interpret them. So this is relevant stuff. All right. So let's say we get these mutants, right? We have 3 different mutants. Our wild type is purple, and of course we know our mutants are going to be white. They're in this pathway. And so how do we know if the mutants are in the same gene, right? So that same pathway here, we're talking about A and B. So how do we know if the mutants are in the same gene or in different genes? And the answer is, we do a specific genetic test called a complementation test. So it's pretty simple. It's basically a test cross like we talked about before. So we're going to cross a homozygote for the first mutation with a homozygote for the second mutation. If the offspring have the mutant phenotype, right? That means when they come out, they're, well, in this case, we would say white, right? Because that's the mutant phenotype. Then they're in the same gene, right? And if they have the wild type phenotype, so in this case, wild type would be purple, then the mutations are in different genes. And how does it work? Well, it's pretty straightforward. So this is just showing you, we cross mutant 1 with mutant 2, and their offspring are wild type. Well, we know that these are in different genes. How do we know that? Well, here's just sort of a representation of this. So here's our chromosomes, right? We have, you got some from dad and you got some from mom. And so in order for this to be homozygous, you would have to have the mutation that you inherited from your father and your mother, meaning this doesn't work. And we designate this 1 because we know there's more than one mutant for this particular allele. Not yet, but we will. So right now you wouldn't have to do that unless you know there's more than one mutant in this. If we had another mutation here, then we would call it A2. I think we've already covered this. And if maybe the promoter doesn't work, well, then we would call that A3 and so on. So even though it has the same result and it's still in the same gene, we have this uh, signify which mutants are different from each other. And we do that by superscript. And then we cross it with a B, right? So mutant 1, this is an A, right? Mutation is an A. Mutant 2, the mutation is in B. We didn't know this till we did the cross. And so because this is homozygous, it has to get one of these. And because this is homozygous, it has to get one of these. So we know the outcome. They're both going to be heterozygous, right? This has to get a big B. This has to pass on a little b. That's all it can. And so in the heterozygote, we're going to get less amounts of enzyme A and less amounts of enzyme B, but that's enough to produce the wild type phenotype, so we would get purple, right? So complementation test would tell us if we have purple flowers from the cross that those genes are uh, different and different genes in the same pathway. Hopefully that makes sense. And then again, this is just showing you um, the designation that we would use. A1 if there's more than one, B1 if there's more than one, and then this is be the designation for the hybrids, the dihybrids. All right, so let's say we do another one. We cross mutant 1 and mutant 3, and the offspring are not wild type, they're still mutant. Well, we know that that means that they're in the same gene, not a different gene, right? These were in different genes, right? So because we got the wild type, this one is the same gene because we got the mutant phenotype. So why is that? Well, we go through our same example, and this is why we have to designate this too, because we know that there's two mutants in the same allele. It's just so this one is at this locus, this one is at this locus, but it's still an A. So it's going to wreck A. It's not going to make A. So this has to pass on broken A. This has to also pass on broken A. So in the end, we're going to get homozygous recessive condition, even though it's two different alleles, but it's still affecting the same protein. So no enzyme 8 is made, and therefore we're not going to get the purple, we're going to get white. All right? So in this complementation test, it tells us that the mutation that we're looking at in those white flowers, because they didn't save each other, are in the same um, the same gene, the same gene that codes for a phenotype. So this is not rocket science. You already know that we can have mutations in different places. So here's the locus for gene A, and this is just the DNA showing you this is another locus. Here's the DNA. So we would designate this A1 and this A2 because even though it's affecting the same product, it's in a different location. All right. So this is what geneticists call gene interaction, which seems pretty common sense and straightforward. So gene interactions occur when two or more genes that affect the same phenotype and a common pathway. And we've already talked about this. This is epistasis. All right. So we don't know if the two genes interact. Um, until we do the crosses. Um, but we can tell if the genes interact even if we don't know the pathway because we do a dihybrid cross between the two genes, and we know from Mendelian genetics that we would expect a 9 to 3 to 3 to 1 ratio in normal Mendel in a dihybrid, let's say this. And you can go back in the previous lectures and look at that. You like if we did the seeds, we did the cross, we would expect 9/16 to have both dominant traits, 3/16 to have one dominant, one recessive, 3/16 to have the other dominant and the other recessive, and then 1/16 to have both recessive. Um, and we already talked about this, so you know that if we do the dihybrid cross in these purple flowers, for example, because they'd both be purple, then we would expect like a 9 to 3 to 3 to 1 ratio. But in epistasis, where two genes are working in a common pathway, we get weird ratios. So we have the 9 to 3 to 3 to 1. And again, this is review. These usually end up together, so we get a 9 to 7 ratio. Or like the example we use for Labrador retrievers, these two get together, so we get a 9 to 3 to 4 ratio. But regardless, these are combining with one another to produce these ratios. And this is just a Punnett square to show that, so I'm going to just kind of graze over that. All right, so a 9 to 7 ratio or a 9 to 3 to 4 ratio indicates two genes are in the same pathway. And like we said, we call that epistasis. Now, we don't know where the genes are in this pathway because either mutation can affect that pathway. So, doing the cross, getting a 9 to 3 to 4 or a 9 to 7 ratio gives us an indication that they're in a pathway. But we don't know which mutants are what in this pathway. And so that's why we do the complementation test. So studying gene interactions, it's basically find two different mutants with the same phenotype, do a complementation test to find out if two genes are involved. If they are, do the dihybrid cross and examine the offspring ratios. So here's an example. We can look at flowers. These are blue-eyed Marys. Wild types are blue. There's a mutant which is pink, and then there's a mutant that has white flowers. Both of these mutations, both pink and white, are autosomal recessive mutants. So we're not talking about any sex-linked things yet. And so we can signify these two genes as, say, W and M, white and magenta. I guess this is what the example is using. So we have mutant 1 and mutant 2, and let's say that the offspring here are wild type. Well, remember, this is a complementation test, and so we know if we cross the two mutants and we get the wild type, that they're complementing each other by providing so this one's providing the W, and this one's providing the M for the heterozygous condition to make it wild type. All right, so then we can do the dihybrid cross once we know that they're interacting in a pathway, and we can figure out what the ratio is. So in this case, we have the 9 to 3 to 4 example of epistasis rather than the 9 to 7 that was shown previously. All right, so now we know it's epistasis. We know there are genes in a pathway. We know which mutant that we have has which mutation and which particular gene. Although we might not know particularly without DNA sequencing where that mutation is. But anyway, regardless, so when we're talking about effects in a pathway, an epistasis pathway, the overriding mutation is what we call epistatic, and the overridden mutation is hypostatic. So in this case, let's say that we have gene W and gene M. So this is going to take white and turn it into magenta, and this is going to take magenta and turn it into blue. So the epistatic gene is upstream from the hypostatic gene. So this, remember, this is the direction of the pathway. So in this direction, we're talking upstream. So the epistatic gene is upstream. This is the upstream gene. So gene gene W is epistatic. And then this is downstream, right? This is downstream. So this is the downstream gene, and this would be M would be hypostatic. So if the upstream gene is knocked out, right, it doesn't matter. We're not going to get the blue pigment. Um, it doesn't matter what's downstream, right? Because this happens first, and everything else is wrecked. Okay. So that's the end of this lecture. Relatively short, 30-minute lecture. Um, I'm going to go ahead and publish this, and then I'll do the. Let's see, what's the next lecture? Next lecture is linkage and recombination. So, yeah, I'll do that as a separate lecture. I might do it today. Probably more likely I'll do it on Thursday. If you guys have any questions, come to office hours. You can email me. I'll see you next time.